production is agriculture, extensive agriculture with annual crops. So truffle production is new and truffle production is starting. But we have several regions in Argentina that where we have critical elements of climate and soils that are suitable for this production. As you can see in this map, we have the red spots with different orchards that we have from Patagonia, from the province of Santa Cruz, that is south here, at more or less 1,000 kilometers, and then Tucumán, that we have also Antonio Posadas, that is a truffle producer that came from that region, and in the different other provinces, and in Buenos Aires provinces we have more hectares planted with this system. We produce truffles in counter season with Europe as Australia and New Zealand, and the fructification here is starting in May and until August. This activity started in 2002 in Chillar in Buenos Aires, and we have planted almost or more or less 200 hectares because we don't have formal registers of the hectares that we have in our country, and more than 40 orchards are present. The commerce is mostly domestic, but we have register of exportation to United States and France. We have one active nursery and 4 or more in development, and the indirect jobs are truffitourism, as is the case of Trufas del Nuevo Mundo in Buenos Aires provinces, and of course gastronomy with the restaurants that are near to the truffle orchards are also in major cities. So in this context, we started to work in truffle orchards of around 20 years of plantation, in analyzing the site conditions and the mycorrhization of the 2 Quercus species, as we have the planted system here in Argentina. And the quantification of Tauber melanosporum in roots, and also the presence of other ectomycorrhizal fungi that act as contaminants. And also if there is any relation with the productive and non-productive trees. We first made a topographical analysis with an UAV technology with these little planes here, and we generate a digital surface model. We made the site analysis, and also with this information we took 24 soil samples in different areas just to characterize the texture and physicochemical properties of the soils. And we observed, for example, that the truffle orchard presents in all the samples neutral to basic pH, a high amount of organic matter, also phosphorus, and these conditions were relatively constant around the orchard. Then we made a design when we observed and we analyzed the roots and associated soil of 168 trees that were in a different topographical areas, high, medium, and low, and we analyzed the 2 Quercus species that were planted, Quercus robur and Quercus ilex. Also, we analyzed productive and unproductive trees with presence of fructification or not present of fructification. We made this with soil cores and also with 20 centimeters of roots of each tree, where we analyze the root mycorrhization. We perform also a sequence analysis with Sanger sequences of the ectomycorrhizal contaminants, and we analyze also 12 compost samples with ITS metagenomics, just to know which is the community that we have associated to this situation. What we saw? We have a strong data dispersion, but we observed that the mycorrhization was lower in Quercus ilex, and where also we could find a scarce number of fine roots and root tips, and Quercus robur exhibiting the highest percentage of mycorrhization with melanosporum. And regarding topographical areas and productive condition, we observed that in the middle and low areas, Quercus robur presents some difference, and in the middle area of Quercus ilex also we observed some difference. And what about the ectomycorrhizal contaminants? We didn't observe a big diversity, but we observed differences between the two Quercus species. And also we observed a low percentage in both cases of ectomycorrhizal contaminants. In the case of Quercus ilex, Telephora and Tomentella were the species that dominate in all cases, where in Quercus robur was Scleroderma, the most frequent. And also, we observe an heterogeneous diversity according to the topography in Quercus robur. We observe in high and medium topography more frequency of Scleroderma, and in the other parts we observe more diversity, but in low frequency. And with this low amount or low percentage of ectomycorrhizal fungal contaminants, we can say that even if the mycorrhization of tuber melanosporum is low, the roots are partially occupied by these contaminants. And what about ITS metabarcoding? The phylum Ascomycota was the most abundant, was the most present, and we found that the host species was the strongest driver of fungal community structure and composition, while production and topographical areas were not significantly affect this situation. This is with alpha diversity. We are conducting now more statistical analysis. And when we observe the relative abundances at species level within the Peciosomycetes, where tuber melanosporum is included, we observe in these pink lines that tuber melanosporum is the main species within Peciosomycetes, even in the two Quercus species, and in the medium slope ectomycorrhizal contaminant. So even in the low mycorrhization of tuber melanosporum present in both species of Quercus, we can say that after 20 years, tuber melanosporum is in the soil, so it's present, and that is a good result for new colonization. And after that, what things we can conclude? As the host species was the stronger driver of fungal community structure and composition, we observed that the temperature, for example, in this place where the orchard is set, the summer temperatures are high and during several days. And this high temperature could affect also the mycorrhization level because, for example, in Quercus ilex, we couldn't observe fine roots or low mycorrhization with tuber melanosporum. So the finest root could be deeper in the soil, and the production of fructification is not very possible in that situation. But Quercus robur that is more affected by